in Paris, and we are going to start the second part of the third session with a talk of Pfaffl about exosome isolation, holistic expression profiling using RNA-seq and RT-qPCR. Thank you, Michael.
Thank you. So good morning, everybody. I hope you enjoyed yesterday's boat cruise and the dinner and the night scenery. I did very well. And I thank the organizers for this nice evening. So let's start with research. And we totally switched topic now. Yesterday we only had clinical talks. Now we're coming a little bit to applied agricultural talk. So I come from Weinstefan. This is a life science center, belongs to the Technical University in Munich, and we are working in cattle and pig. And this will be a talk dedicated to cattle, milk, blood, exosome, microRNA, networks, and regulation. But the first slide will be about the challenges in diagnostics. And this is overall species, molecular challenges for the future. What do we want? Definitely, we want to test our markers in a minimal amount of tissues. That's why we are here. We are talking about microgenomics. And this should go down to biopsy, to sorted cells, or even to a single cell. And therefore, if you have less amount available, we should have a very sensitive assay. And this should go down to single molecule detection. Everything should be fast and reliable within 1 hour, and therefore we can detect early any kind of diseases or microorganism in food or biomarker or even biomarker pattern. And we should be able to multiplex, because maybe one biomarker is not sufficient, or multiplexing due to a set of microorganisms. On the other hand, we need a wide quantification range. Nobody knows before how many analytes are in my biological sample. Are they single molecules? Are there 1,000, millions, or even billions? Therefore, if you're working with standard curves, we need a wide standard curve. And we need a stable assay which is highly standardized and reproducible to measure this so-called biomarker or biomarker signature. Brings this to an end, meaning PCR is a very good method to detect these, to measure these, and you also can amplify, analyze, and quantify in one tube. The more steps you have, you know it from the labs, you're losing material, even small amount of analytes attached to the cell wall, and they are gone. And if you do everything in one tube, then you're not losing any material. And in terms of PCR, highly standardized, we are talking about the MICI guidelines. On the other hand, if you're looking for biomarker, we need new screening technologies. It used to be array hybridization array. Nowadays we have better tools, more sophisticated tools like RNA-seq. And therefore we can define, find, elucidate new transcriptional biomarker. And this is what's the talk about the elucidation of new transcriptional biomarker pattern. And if you look in the literature, we've done some microRNA profiling, so microRNA in exosomes are well established now in the literature, and if you're looking on Medline, microRNA publications and especially exosomes are increasing exponentially. Therefore, exosomes are known to be the new biomarkers in any kind of tissue. Just put some slide on, some reviews about prostate cancer and let's say colorectal cancer. Okay, how does these microRNA work? I think I don't have to explain it to you. They were expressed from genes. Mainly this is the best known pathway. And then they bind either single microRNA to a lot of messenger RNA, and on the other way around, a lot of microRNA to a single mRNA, and then say either, if they're perfectly matching, say cleave these mRNA, or if there is one mismatch or no perfect matching, say inhibit the translational repression, mean they inhibit the translational process. And to show a slide about the capacity in milk, where we are working on, There was one paper published where they had an artificial knockdown with artificial microRNA. First they did it in cell cultures, then in mice, and afterwards in cattle. So you can knock out single genes. In this study it was a beta-lactoglobulin gene because it causes a lot of allergies. So in the end we developed a kind of workflow in our lab where you have a model organism in ours. And there are cattle, can be human, it can be mice, where we're going for transcriptional biomarkers is our approach. There we have, on the one hand, we have the mRNA, we have the microRNA, or we have other small RNA available, or let's say long non-coding RNA. We can screen them with RNA-seq or with microarray to have an independent, holistic tool. We have to verify this. Of course, we do this with RT-qPCR. And then at the end, you have biostatistical tools to validate your biomarker or biomarker sets to come to your significant biomarker pattern. Okay. So now I want to lead you through several studies we've done over the last year. As I said, we are working in cattle, and the first thing is, what is the biomarker pattern? What is the microRNA pattern in milk in comparison to blood in bovine? Nothing is known about it. Therefore, we compared both annelids, both tissues, in a colostrum, early, mid, late lactation, to have a little bit of influence on the lactation stage. And in parallel, we sampled blood from the same cattle. And we quantified them, or we screened them via small RNA-seq. And afterwards, we confirmed our results via PCR. What we want to predict, we want to find functional microRNA regulating the mammary gland or the blood. We found to find the mRNA targets, which will be targeted by the microRNA, the pathways behind it, and we call these then integrative analysis of mRNA and microRNA. And what about the function? This is not working here now. The function, we are interested in the mammary gland physiology, definitely mammary gland immunology. You know, mastitis is a big problem in agriculture, and we want to know more about it. And on the other hand, we have here regulative microRNA, which are present in the milk, and therefore there might be an effect on the newborn calf via the milk on the first day, weeks, and as well maybe on the consumer, because we are also drinking lots of milk. So we set up a study where we sampled from one cattle, blood sample, milk sampling, always in parallel. On the colostrum, we call it days in milk 1, the early lactation, the mid lactation. Here you have the lactation day, and the late lactation. So cattle gives around about 1 year milk, about 305 days. And at the end, we ended with 2 tissues with 4 time points and 3 animals. So we had 24 samples. Then these samples underwent, of course, centrifugation to defattening the milk, and then we had, for this study, we just extracted total RNA out of this defatted milk, so it's total RNA out of total milk minus the fat. And later on, we're coming to subfractions where we have, of course, differential ultracentrifugation, but this is later on. The first result is very surprising. Here we had the milkings from the first, the second, the third, the fourth, and let's say a summary of all the remaining milk from all the other 305 days. And you clearly see that the colostrum, the first milk on the first day, is full of RNA if you count it on a milliliter of milk. If you compare this in our studies, this was a separate study, you have here colostrum, early, mid, late, you see that's about 7-fold more RNA in this milk, and in blood it's more or less constant over this time. Then we took these extracted RNA, of course after integrity measurement and all this stuff, and sequenced it. So we had 24 samples. We put 2 times 12 samples on an Illumina array. This was done together with Vladimir Binic at the Amble. And afterwards, since this was not the first study, we were able on our own to analyze these RNA-seq data. And we had two kind of analysis lines. First, you always sequence, then you have the quality control, meaning the Phred score tells you about the quality of the sequencing. Afterwards, you go for trimming, and this time we had 12 adapters, which we had to trim away. Afterwards, you map, and this is a big problem if you're talking about cattle. Mapping genetic information, you can map against the bovine, fine for genes, fine or half fine for microRNA, and not very fine for any other new candidates. So often we map against the bovine gene or databases, and on the other hand, we have to map against human mouse or rat, where much more data is available. In terms of microRNA, to have a guess, in humans, around 2,500 candidates are in the database. In bovines, around 750 to 800, depending which database you're working on. So it's threefold different in candidates. Then you annotate, and at the end you can calculate differential expression. We've done this, so we had the Illumina HiSeq, the FastQC, this is identical, and then we have a very nice, convenient black box, but it's a very nice program. You put the data in, press the button more or less, millions of options definitely, but then you get your data out, and these data are compared to a big database of all small RNA available of all species. So you have microRNA, PEI, and viral RNA, SNOT, and so on, of plant and whatever. And on the other hand, you can more dedicated, more focused analysis with B-trim, bowtie, and so on. So this algorithm are the best adapted, I have to say, or most common one, where you can compare your data with either the bovine or the human databases. Okay, this took, coming back to this, even the development of these new analysis methods for bovine and to all these tryouts took at least 1 year to be confident which data we're working on and which gives the highest confidence. So coming to a little bit of results, if you know about RNA sequencing, you should know these numbers. If not, we had about in this small RNA seq about 17 to 20 million reads per sample. The read length was 52 bases. And if we compare just as a screenshot here blood, you clearly see that the microRNA is the biggest proportion. And here you have the major, or here's some hairpins, some pre-microRNA are also present, and you have a big proportion of this called piRNA, and as well SNOR and the wall, at the end as well viral RNA present in the blood. And if you compare this now with milk, it's a little bit different. You have as well microRNA, but the much bigger portion also called piRNA. So in milk, it's quite different from blood, and milk is an own tissue, and it's synthesized independently. It's not that everybody assumes here in the room everything is in the blood, it's going directly to milk. No, it's wrong. You see, even here in this microRNA pattern, the milk is synthesizing its own small RNA pattern, meaning the cells are coming into the milk, but then they change the physiology. The white blood cells coming into milk called then somatic cells, and of course we have epithelial milk cells doing their own work. So two different profiles. And if we do it a little bit more convenient plotting, you see here in blood we have one big peak around the microRNA, and here in milk, here the different or the individual samples, we have the so-called piRNA. Next question, what are piRNAs? Why they are in milk and why they are so dominant compared to blood. So go on the web, look for databases, and there is only one database available. This is an Indian database, and if you look here, you have huge numbers. Let's concentrate on the last line. Total position mapped to the genome for PI. Wow, these are 100,000 of possible gene locations allocated to piRNA. But this is our multiple locations, multiple loci. Therefore, we have around about, in human up to now, around 20,000 different piRNA. And in mouse and rat, we have around 35,000. And interesting, in bovine, we have no single information about piRNA in bovine. Nothing is reported up to now. So you see, it's about 10-fold more candidates of PI compared to microRNA. The next question is, how does this piRNA work? And here we have the classical pathways for a microRNA with dicer, with X14. Then it gets processed by dicer, and we have translational repression or activation. We know these. And if we compare these to piRNA, more or less the same kind of molecules, a little bit longer, 5 to 10 bases longer, but these are expressed in clusters on the DNA. They bind to so-called PV proteins. They called P element induced by wimpy testis. Why testis? Because testis is the only tissue where they are deeply investigated up to now. They have a dicer-independent processing, and at the end, they interact with the agoproteins compared to the microRNA. This is comparable, but what is the function? So as well, a translational control of the mRNA. As I said, they are investigated and tested. Therefore, up to now, only germline transposon silencing is reported. And if we are talking about transposon silencing, you see they are acting not only on the mRNA, they're acting as well on the rearrangement of the DNA in transposons. Therefore, it's assumed that they have an epigenetic regulation, an epigenetic function beside gene silencing and retrotransposon silencing. And then I looked a little bit, you always find this new information, you go deeper and deeper and deeper, and what you find is, I didn't notice this, I didn't know it, that in humans and mouse, and possibly as well in bovines, we have about 40-45% of our genome is so-called transposomal elements. So a huge amount of our genome is rearranged, and these guys, these piRNA, they regulate this stuff. Therefore, this is kind of very important, I think. Okay, and also in this publication, there's a very nice slide. So you have these transposomal elements, meaning DNA, which is rearranged. And of course, if you change the DNA, you change the chromatin structure, you change gene expression, and you possibly change the phenotype, and so on, and so on, and so on. And this is this story. But up to now, even in humans, very, very less is known. All the publications now are reported in testes, and you nearly find nothing in other tissues. So we were interested, therefore we made the first data. We had a lot of data, of course, you see it, and we made the first comparison between milk and blood, and you clearly see that milk and blood is quite comparable, but here days in milk 1, meaning the colostrum is totally different, even compared to milk. So these colostrum exhibit shows a very, very individual expression pattern for this piRNA. Any kind of pathway analysis, functional stuff is not available. There are no database available. And we are now on the way to validate these candidates which we found in here, but no even assays are available. So we're working together with QIAGEN and Exicon, developing new assays. We have to design the assays according to human, mouse, and rat sequences. Of course, you can imagine that it's not always working, but we managed this to validate at least a 30 high abundance piRNA. In terms of which genes they target, what is the physiological function, only question mark. Very less is known about it. So, as a first summary, these piRNAs are regulative RNAs compared to the microRNAs. Seems very interesting for us. Why they are so prominent in milk, we don't know. Why they are so prominent in the colostrum in the first milk, we don't know. To summarize, they're a little bit longer. There are around about 30,000 candidates in human and mouse. In bovine, we don't know, but we assume it's the same amount. They are expressed in clusters. They show much complexity because they have a low sequence conservation. I already said this, and at the end, compared to other small RNA that is involved in silencing mechanism, but the pathways are poorly understood because they are primarily investigated in the tested and in germline cells. So we concentrated on the microRNA because Therefore, a lot of information is available, and here we were working with a Mirbase 19. This was a database from last year. The data are from the last year. And therefore, we had, during this time, 755 microRNA candidates in bovines. And you clearly see, in milk we found 566, in blood 582. And these are the microRNA numbers which are not changing by number, and you clearly see day 1 to early, mid, late, the expression pattern of this microRNA is changing over time in milk and in blood as a pattern as such. And if you make a principal component analysis, you see that the blood as such is very stable, independent of the year of the lactation time. You see here all blood samples cluster on one spot. Here you have a huge difference between the milk, and interesting, these days in milk, these are these light blue, they are also very different from the rest of the milk, meaning milk and blood is different, and colostrum is also very different from the other microRNAs found in normal, or let's say table milk, from early, mid, or late lactations. And if you plot it, you clearly see, again, here you have the blood samples, here you have the milk samples, again, clustering of the 3 colostrum samples, and here you see the microRNA candidates which are responsible for this separation. So I have here prominent, like 148A, or in blood, like 451, those is somewhere out here, though it's a major marker, and a lot of others. And afterwards, we showed, we compared, we always go for the highest, 30 highest, because this, you can visualize this very nice. You can also go for the highest, 50 or 100. And you clearly see blood is totally different from milk, and somewhere in between you have a cluster of the cholesterol days in milk 1. If we are looking now on the significantly differently regulated genes, let's start with blood. We see that over the whole lactation process, of the difference from day 1 to early, from early to mid, from mid to late, there isn't that much changes. But if you look in the milk, you clearly see that the regulation of the microRNA, or the regulatives, the significantly different expressed, are very different from the colostrum to the early, and then it more or less keeps stable. Therefore, we concentrated, because we are interested in milk, on the difference between colostrum and the rest of the milk in the remaining days. And these are the cluster analysis. I skip these, and I'm coming to that we also validated, of course, our results, because NGS was new to us, and we validated all these candidates in bovines. We designed our own assays, and it's always interesting, does the RNA-seq data fit the gene expression data? And you clearly see here you have a very good correlation that in terms of data fit, and it means we can trust RNA-seq data, we can trust quantitative RNA-seq data. Okay, the next question is, what does these majorly expressed genes and significantly regulated genes in milk, in colostrum in comparison to milk, what does they do? Therefore, you have to take this data, import it in so-called pathway analysis software tools, either the target base 6 we used. They have 65,000 interaction. They are known to be validated, and on the basis of CAC pathways, they give you the result which target genes might be regulated. We confirmed this as well with the Diana package, and as well this is a package in these Genomatics software tool, and we ended up with the same pathways. So the significantly regulated genes of differences from colostrum to milk are responsible for cell adhesions, means cell matrix adhesion, integrins. They regulate these kind of mRNAs, and afterwards hopefully as well the proteins. Extracellular matrix interaction, mTOR signal pathway, and Also, the colostrum is known to be like a wonder milk, and it's supported in the internet that you can cure cancer. And interestingly, if you import your microRNAs in these databases, and out of the top 10 pathways, you get 7 cancer pathways, where we found that these cancer-inducing genes, or cancer-related, sorry, is a better word, cancer-related genes are highly regulated by this candidate in colostrum. Good. To summarize the small RNA story, we found differences in milk and blood. We discovered new microRNAs. We discovered definitely new piRNAs in the mammary gland and in milk as such. We have a higher microRNA dynamics in milk compared to blood. And these microRNAs might be, I will say, conjugative in cell adhesion, in mTOR pathway. They are induced in various disease and cancer-related pathway, and I didn't show these results in immune cell differentiation. So for us as agricultural interested or veterinary interested, we think they influence the interaction between the pathogens and the mammary gland. They due to the immune cell, impact on the immune cell, they are responsible or regulate the diabetes and the immune status. Due to the mTOR pathway, the epithelial cell renewal in the mammary gland, and of course the milk synthesis, because we have these differences between colostrum and the rest of the milk. And also we think about the microRNA transfer via the colostrum to the newborn, and also about the regulation of the newborn calf in the first day. So we hope to have new insights into mastitis and immune imprinting, because on the first day they get this milk and kind of imprint the young calf. So, and you end the study and then you end with a to-do study. So the to-do list is revalidating, this is already done, and then I highlighted it, where does these small RNA are coming from? We just analyzed the milk, we just analyzed the whole blood. Are they coming from the cells itself, in the blood, in the milk? Are they circulating? There's a lot of paper about circulating RNAs now. Or are they covered or stabilized in so-called exosomes? And of course, we want to describe them functionally. This brings me to the last part of my talk, where we are looking where this microRNA information is coming from. Not just from milk or blood, so we want to fractionate it. And therefore, there are 3 possibilities. They are intrinsically stable. That's what some people said. They are short mRNAs with a length of 21, and therefore they are not degraded by RNases. No, this is not true. So most of them are bound to proteins and therefore stabilized, or they're protected by vesicles. And there are some studies out. Some microRNA maybe focus on vesicle stabilization. Others are maybe bound to proteins. So the next story which we have to test, are they protein bound, or they are in vesicles. And if we're talking about vesicles, sorry, first we have to place this study. The next step we've done was, can we isolate microRNA from plasma, meaning from blood, from out of cattle plasma in a minimal amount of tissue? So we optimize the extraction for circulating small RNAs from plasma, cattle, to investigate these microRNA and piRNAs. This was a study done by one of my PhD students, Melanie Sporcraft, as well in collaboration with the EMBL, because they have the very good sequencing facilities, and we are just on the way to publish it because it's already resubmitted. Some slide out of this study. We started with 1 milliliter, 6 milliliter, 9 milliliter. We tested various kits, bead system, whatever, and we ended up that we need at least 9 milliliter plasma to isolate small RNAs. Here you see the bioanalyzer runs from A or B. There's nothing, and with increasing volume, So we worked with this vacuotaneous system from QIAGEN. We could isolate small RNAs, and this is a slide after library prep for small RNA sequencing. And you clearly could see that there are already 2 peaks here in the library. There are 2 lengths of small RNAs majorly present, and you can think which kind of RNAs these are. This will come in the next slide. We are talking here about 9 milliliter of circulating total RNA. We are talking here in the range of 15 nanogram. And we sent these 15 nanogram for RNA-seq to Vladimir, and he said, no, it's too less. We never made RNA-seq with 15 nanogram. We always want 1 microgram or 500 nanogram, but not 15. And he said, let's go for it. And at the end, you see, we got lots of reads. So in average, around 10 million reads in these 50 nanogram. So it's a miracle. You don't need that much, even with very low amounts and a good procedure, which is definitely given at EMBL. You can sequence and get a lot of information which fit to the database. But this is another study. I just want to show you that it's possible to isolate out of minimum volume of 9 milliliter, huge amount of total RNA, which is suitable for sequencing. And again, you have these 2 peaks in the library break. And again, after data analysis, you have, this time in this study, we found much more piRNA in the bovine blood compared to the other study. And if we compare it to databases, this is a significant difference between microRNA, the amount of microRNA candidates compared to piRNA. So, the next step was given. We have this stuff in plasma, but whereabouts in plasma? So we went one step further on. Are these microRNA circulating in the plasma, or are these covered in exosomes? So the next step is we are talking about microvesicles in tissue, here in blood, in biofluids. We have apoptotic bodies, which are much bigger. You see here, they are in the size of about a micrometer range. We have exosomes, which are very tiny. We are talking about nanometer range, 40 to 90, 40 to 100 nanometer, and also other shedded whatever vesicles. And I think it's still challenging now to say, is it an exosome, is it an endosome, is it an apoptotic or a shredded or whatever microvesicle? Because we are working in the or in the nanometer range. And what we did, we took our samples, we made really lots of preparations, ultracentrifugation, magnetic isolation. We added, we combined them, and at the end, we made some electron microscopic staining, and we assume that these guys, these very nice round, in all papers they are round, I think these can be exosomes, and you have, some stick together, and on the other hand, you have also like fragmented, this seems to be more apoptotic bodies. Okay, what are these guys, what we call exosomes? They are classical membrane vesicles, the size about 40 to 100 nanometer. They have surface proteins, they have internal proteins, they have DNA, mRNA, small RNA, microRNA. So we focus on the microRNA. They are secreted by various cell types, almost all the people assume, and they are very stable. They are pH stable, they are stable against enzymes, they are very stable in storage, and they are resilient against freeze-thaw cycles. And to say some word about the secretion, so here you see a cell. The first is that you get a cell membrane endocytosis, and then you get endosome, and this endosome interacts with the endoplasmatic reticulum and the Golgi apparatus. Therefore, at the end, you get multivesicular endosomes, or also called intraluminal endosomal vesicles, and this will be then shielded off the cells, so you have exocytosis. Therefore, you have these small, so-called exosomes in the size of 40, 50 nanometer with a cell-specific information. So the theory is that every exosome coming from different tissue have a different content and as well have a different cell membrane, because the cell membrane is part of the exosome. So, some screenshots from the literature, exosome content. You see we have surface proteins, we have mRNA, we have DNA, but we have a lot of candidates of microRNA. If you go to other paper, this is a service provider which helps you to purify them. Here are the major target genes. We are also working for, or worked for purification or for surface detection, for identification with these so-called tetraspanines, CD9, CD36, and CD81, to identify exosomes from as different from, let's say, apoptotic bodies or other microvesicles. Okay, we've about one word about the function. What is the function of these exosomes? They have extracellular proteins, and they are full of microRNA. The first is a juxtacrine function. Juxtacrine means cell-by-cell interaction, like a T cell is activating a B cell, cell-to-cell interaction, or there might be a cleavage of the extracellular protein activating a transmembrane receptor and have a signaling, or what we more or less favor is that these content in these microRNA is released in a target cells, and these microRNAs in these exosomes regulate the so-called target cells. So we are calling about a hormone-like communication, not like a single hormone to a single tissue. Here you have full of regulators, and at the end I think it's a mixture that you have an income and surface receptor binding to a tissue, and then you have a fusion of your content with the target cells. Good. Some papers are already out from 2012. This was more or less the start point to research as well with exosomes. Bovine milk contains microRNA and messenger RNA. This is stable. This is also reported for human breast milk, and these have a function. Okay? Good. So our exosomal workflow is, and we are still in the very beginning, to optimize the isolation from blood and milk and other matrices. We have collaboration with urine or with other institutes working on urine or cell culture. But still is a problem. What's the exact size? You cannot do with every extraction electronic microscope pictures. This is far too complicated and far too expensive. Therefore, there are some very nice machines out which scatter laser light, and therefore you can see the concentration and as well the size determination. So they are also available in different sizes coming from one tissue. And next step is the biochemical characterization. What is the content? The mRNA, the microRNA, the mRNA gas. Then we have surface proteins and intraluminal proteins, maybe remaining DNA, and very interesting, some people also pronounce that the exosomal cell membrane composition might be different from tissue to tissue where they're coming from. And this has also an impact of these exosomes, how they fuse to the target cells. So we are interested now on the transcriptomics, on the genomics, on the proteomics, and as well on the lipidomics of these exosomes. They have a hormone-like function, and we want to know what is the regulatory function in these body liquids, milk and blood, mainly on the immune system or cell differentiation in the target, either in the calf or maybe in the milk for humans. We drink a lot of fresh milk, and there are some ideas out that fresh milk might be an influence on children and on the asthma risks. So we also have another project researching this. And of course, if you're doing all this stuff, you need a lot of bioinformatics. You need young and good students working with our packages, post-translatory regulatory networks, interacting different omics data from mRNA, microRNA up to proteomics and lipidomics. So let's come to our results. As I said before, first we're working here with milk. First we have a defattening step, and then we have differential ultracentrifugation. This is for blood different than for milk. We have between 5 and 7 ultracentrifugation steps. The last step is 100,000 G for 2 hours to plate these exosomes, and then they're coming on a sucrose gradient, and between 30, 40, and 50%. We used to have 5 steps from 20 to 60. But we know now that the milk exosomes accumulate in the middle layer at around 40%. This is a density of 1.18. But the density, you see it here, is a little bit heavier than water. It's 1.2, 1.18, something like that. Okay. Then we started to characterize these. We've done this always in parallel from milk to blood because we want to know, are they coming from blood, the exosomes? They're small, they can go through the mammary gland epithelial cell. Here you see the CD63 is an exosome, a general tetraspanine exosome marker. You have it available in blood and milk, and as well in a glycosylated or dimeric form here. And here we have a milk-specific surface marker called MFGE8, which is only present in milk and not in blood. So there's the first difference, that milk exosomes are different from blood exosomes. Therefore, they are synthesized in the mammary gland, either by the mammary epithelial cells or by the somatic cells in the mammary gland. Coming one step further on, if we isolate total RNA for the transcriptomics, if we're taking here milliliter blood, we have lots of mRNA. As I showed before, colostrum has about 7 times more than traditional milk. This is total concentration in blood, colostrum, and milk. But if you isolate exosomes and isolate RNA, you see there's nothing left. So in blood, there are very less amounts of exosomes compared to milk. Classical milk. Milk is full of exosomes, and again, the colostrum is hyper full of exosomes. So you have here a range of 20, 50-fold differences in total RNA from colostrum. So there must be something, there must be an action, there must be a function on the calf. Okay. These are the first results we received a few weeks ago. So you see here that we compared 3 cattle, the exosomes from 3 colostrums, exosome 1, 2, 3, and this is very comparable to the total colostrum, meaning we could, up to now we couldn't find a real difference in pattern. One milk we have to discard because the RNA sac was inefficient, and if you compare this to colostrum from the first study I've shown you at the beginning, these are highly comparable, meaning colostrum as such has a stable and individual microRNA pattern, which is totally different from your drinking milk, what you drink or what you have on the table. Okay? This is huge. This difference is a huge difference on this cluster. And if you can plot it as well in a 2-dimensional way, you have here the colostrum, the exosome, the other colostrum, which have a subcluster in differences to other milks. If we compare the 30 height expressed genes in these, it's a little bit difficult. In general, the pattern match, but here you see that some cattle or some markers are present or not. And this is what we try to find out. Do we have markers for exosomal milk RNA and exosomal blood RNA markers? Okay? But these results are very fresh. We have to validate these. But there are a lot of markers, even of the highly expressed genes, which are identical, but some are totally different. Therefore, we have individualized microRNAs from cow to cow. And then we compared this. This was a study where we had blood and milk from the dams and only blood from the calves, from the own calves on the first day, because we want to see, is there a takeover of the exosomes through the mother, through the cow milk, to the colostrum, to the calves. And these are very new data from last week, and we didn't have the time to analyze them, but you clearly see that the dam milk and the dam exosomes, these are the same study, are highly comparable, meaning the exosomes and milk are identical, that the calf and the mother's blood is highly comparable. So this is due to the blood, and there is a huge difference between milk and blood. But if we find a pattern over the days, let's say, a pattern from the first hour, the third hour, the sixth hour within the first day, which is influenced by the first day colostrum. This one we have to go much, much deeper, and I don't have the data up to now. Okay, to summarize, these exosomes finding what we have up to now. First, exosome isolation is very complex and very time consuming. Up to now, the ultracentrifugation is the best method, but you need a UZ, and this is a very heavy and time-consuming thing. All the centrifugation takes you at least 1.5 days, but it gives you much, much better quality compared to any other kit called ExoQuick or something like that. QIAGEN and Exosome are coming out with new kits. We didn't test them up to now, but we will definitely, we will. Exosomes are highly abundant in milk and even higher abundant in colostrum, low abundant in blood. This was also a finding. We have differences in the RNA spectrum. We have mRNA, I didn't mention, we also have piRNA up to now. There is a different spectrum, colostrum versus milk, and milk, of course, versus blood. And the total milk and exosome transcriptome is highly overlapping, but we have single markers which are different. The to-do list, of course, validation of these candidates. Are they there? Are they not there? Find stable and significant expression signature in exosome, in milk, describe the function, and better characterize the protein and lipid level. This is all done, or hopefully done, in the next PhD started this year, and at the end, integrative bioinformatics. If you want some more reading about transcriptional biomarkers, we edited with the last meetings in Freising 2 methods issue, where you find the last about the evolution of qPCR and the transcriptional biomarker. And we also had all the talks recorded from the last 3 meetings. There are 200 talks about PCR, NGS, and all this stuff. This is a free web page called econferences.de, and you can watch all the movies from Michael and Ken and Dave and all the qPCR community. And at the end, hello, I have to acknowledge my 2 PhD students which are majorly did the work. It's Benedict and Melanie, of course, the Amble team, Vladimir Benisch and co-worker, and as well the Genomatics team, because we're still developing the Genomatics software to be better. And I thank you for your attention. Thank you very much.
Thank you, Michael. Very good talk. Questions?
Thanks. Do you have variation in microRNA content in cattle populations? And does it correlate with the milk quality, whatever you call quality?
No. This is the results from another study. As I said, we're doing another study together with human clinicians from the pediatrics, and we sampled milk from, I have to think, 3 different farms in Bavaria, 3 different breeds. This was Simmental, this was Brown Swiss, this was Holstein Friesian, and from 3 different cattle. And we looked about the microRNA pattern as such. They are highly comparable between different feeding regime, different cattle. Yeah, there are differences, but not that much.
Michael, a very basic question.
Thank you.
How can you find pVRNA in cows? Because I'm assuming that not even the Munich cows treated with all the testosterone do have testes.
Once again, no, we compare it.
No, but
with
a cow does not have testes, and don't you need pVRNA for?
Now I understand. Yeah. Yeah, up to now it's reported in testes, you know, but there are definitely.
Recently, do you know if the microRNA in the exosome have an impact in the target? Do you think if target the cell of the digestive apparatus of the young cattle, or we can find it in the blood of the young cattle.
That is this study. What we are assuming that we have lots amount in the colostrum. And as you know, maybe some of the human clinicians did not know or does not know it, cattle transport their antibodies to the offspring via the first milk. So they have a very complex placenta. The antibodies cannot be transferred from mother to calf. Therefore, they transport the antibodies via milk. And therefore, and this is our theory, the tight junctions in the gastrointestinal tract are open on the first day. That's why the colostrum is full of antibodies. We found, of course, they are also full of exosomes. And our theory is that as well as the regulative exosomes are going to the open tight junctions into the calf. And that's why we made this study where we compared the blood and the milk of the dam with the blood of the calf. And this means here where we have it 0 hours means this is the calf blood before the first milking, okay? Therefore, the theory is to have it here after 1 hour, after 3 hours, and here we fused 9 to 12 hours to find markers from the dam milk fraction in the calf blood milk fraction. But up to now, with this kind of analysis, it's too flat, you know. You will not find the right marker. We have to go much, much deeper into this data. And we have in this study around about 8 million reads of microRNA, and this is a lot of work, bioinformatic work, to find maybe a marker which is different from calf to cattle. And but this supports the same idea you have. Is there a transfer? Is there a transfer of information, a kind of epigenetic imprinting, epigenetic programming through the food to the calf? Right. We don't know up to now, but we hope to find. Please.
Can you label the exosomes so that you can put them in the calf and follow them? And do you envision a therapy in which you make artificial exosomes and you feed them to the calf?
Right? That's what we do. We want to find them, we want to label them, resurface, so we have ideas to make like fatty acids with fluorescence labels where the fluorescence label is inside, and after fusion you have then a positive cell, a positive tissue. One thing, we're working together as well with clinicians from Munich which want to separate or want to knock down in patients some tissue, so they have it's from the cardiology. They want to knock down some cardiology genes, and they're working up to now with adenovirus, and as I said, use exosomes, and they use a combination between exosomes and magnets, or magnetofection they call it, magnetofection, where they put like, or that's what we want to do, like a magnetic coupled microRNA into exosomes, and then the patient will be laid under a magnetic field like we've seen yesterday, same idea, and then they release this kind of exosome in this tissue. And something like that is thought about in Munich in the Rechtsdays. Yeah. Very hot, I think. Please.
Just surprised that the experience in the last 100 years in which people were fed since very, very young, breastfed, they just, well, I mean, everything was fine. So people just developed normally. So, and there they were fed with cow milk, humans. So what would be then the real impact of that?
But you possibly know from the human clinicians there is a complaint from some of them about milk, that milk is bad for cardiovascular diseases, for obesity, because adults normally don't drink milk. We take a lot of fat inside. So there's always a complaint about the health impact of milk, not on children, but on adults. And therefore it's heavily researched up to now. And the industry is very interested about new kind of regulative components in the milk. Yeah. And there is as well another study, so the last comment, I have to stop. There is one study where they tested the impact on milk on the immune response of kids. So kids got different types of milk. Different types we have? We have the fresh milk, the raw milk, and then we have homogenized, pasteurized, ultra high temperature treatment. So we have more or less fresh milk, a low temperature treatment milk, and high temperature treatment milk. And interestingly, we found that they have different microRNA pattern. Different milk, different processing. The processing of the milk have an impact on these regulative microRNA. And if you load these microRNAs, these candidates from these different processing steps into your TAR base, you interestingly find that this is correlated with immune induction, interferon gamma receptor, interleukin, and so on. So there is a connection between milk processing and maybe asthma and immune status of the kids. And the first idea was of this study, is there a difference between the city children and the children from the countryside? I think this is always known why they are more stable against, let's say, asthma or any allergic reaction. And they looked about proteins and blah, blah, blah. And now we are down to the microRNA and to the exosomes. So there might be something. So we are testing it, or we have cohorts where we're working together with a human clinician. We have already cohorts. We have the milk they drank, raw, pasteurized, homogenized, low heating, up to 3 minutes heated on your own oven, which is the worst. And we have the phenotype of all the kids, and we have blood of the kids. Therefore, we can look now for our candidates, which microRNA we discovered in the milk, and we can look in the blood of the kids which genes are regulated. That is fit together. And this would be perfect. But this will be done in this year, or in summer, or by end of this year.